I sometimes feel like sterile technique is one giant game of the floor is lava, except so is the table surface, your hands, and even the air. We're here at Bio 1310, and I want to talk to you about sterile technique. The first step is to glove up and clear your desk of totally normal things. Afterwards, I'm going to use ethanol to sterilize the table surface, but honestly, any chemical cleaner will do. You'll notice that on all your items are these weird zebra stripe tapes. This is called autoclave tape. An autoclave is an oven sterilizer. And when properly sterilized, this indicator tape will go from white to the zebra stripes. Contamination often comes in the form of mold, so check your solid or liquid media to make sure that there's nothing sketchy growing inside. Everything here looks good, so we're ready to rock. Inside this conical tube is something called LB broth, which is basically liquid food for bacteria. I'm going to transfer some of that into the small tube in front of me. So I'm going to load a pipette tip onto my pipette, and with a little bit of dexterity, I can unscrew and hold onto everything like this. Remember that the table is technically sterile from the ethanol, so you can put the cap down, but this is just a bit safer. Now I'm going to replace the cap and then switch over to the micro centrifuge tube. Pop the cap off with your thumb and begin the pipette transfer. Notice the entire time that I'm making sure to close lids and keep the pipette tip vertically suspended as much as possible because everything is lava. Once done, I eject the pipette tip into the waste container. There may be times when you have to pipette out of a jar like this. In that case, I can sterilize the lid using our best friend fire. Here I'm lighting the candle, unscrewing the cap, and warming it over the flame once or twice, being careful not to melt the plastic lip. However, you don't need to sterilize the tops of ethanol jars since they aren't capable of growing mold anyways. Solid media is called agar plates. They look like this, with two plastic lids sandwiching a jello surface inside called agar. Collected bacteria grows on this jello surface. Labeling always happens on the back side only since the lid can swivel and fall off. Let's say I collected a sample on the ends of these cotton swabs and I want to transfer them onto the agar surface plate. I'll take out the sample swab and then place the agar lid face down onto the sterile surface. With the swab, I'm going to try and color the entire surface, turning the plate around if I need to. When I'm done, I'm going to transfer the sample into the waste container and then recover the lid. By the way, total bragging rights if you can do all of this one-handed, but it helps to have a partner. When transferring a bacterial colony onto another plate, we simply take a sterile swab and tap it on top of that colony, and then repeating the same streaking process as we did before. Be gentle when using your swabs, otherwise you might end up ripping the agar like this. Another pro tip when streaking plates is to use the light reflections to check for any missed spots. It looks like I missed a few spots, so I got some work to do. Now, what if I want to transfer a liquid culture onto a solid surface? I'm going to remember my sterile pipette skills and then go ahead and load a tip onto the end of my pipette. Then I'm going to take in some of that solution and then pipette that onto the agar surface. As usual, work quickly to make sure that we keep caps covered and making sure not to touch any surfaces accidentally. You'll notice that the bacterial solution doesn't exactly absorb into the agar. In this case, we're going to use this glass hockey stick thing that we call the glass spreader. To sterilize the hockey stick, we'll bring back our candle flame and a small jar of ethanol. I'm going to dip the end of the hockey stick into the ethanol, just enough to coat the end. And then I'm going to safely burn off the ethanol by sweeping this end across the flame just once like this. We don't want to hold it over the flame, otherwise you'll end up burning the bacteria that you're trying to spread in the first place. Know that flaming ethanol still drips, so remember to clear the area of any paper or hair and to hold the stick so that the flames don't accidentally dribble down into your hands or the ethanol jar. If you're transferring from one sterile source into another like this, you can actually reuse the same pipette tip. After all, nothing is technically cross-contaminating here. But if you're not sure, always grab a new tip each time. It's better to be safe than sorry. In the back are the biohazard cans that you can open up with a foot pedal. The general idea here is to toss anything that has living things that could hurt someone or potentially grow anything that could hurt someone. Sterile technique takes practice, so remember that everything is lava, and if you share anything, trust nobody. Good luck out there.